Okay, so when you get signed in, if you can go to obviously Google Classroom, go to Science 10, okay, period 1, and you'll see that there are 2 new things in the stream. Okay, one is our Properties of Compounds lab that we're going to be doing on Monday, and maybe even starting some of the pre-lab stuff today, depending on how quickly we get through the lab report stuff. Okay, and then there's this one here, lab report expectations and format. That's the one we need to have open right now, so if you can just click on that. Okay, it'll have a Google Doc over here in your work. Okay, that's what you need to have open, as that is what we're going to be working through. Okay, so you can add to this or highlight things as you see fit, whatever it is you need to do. Okay, but the purpose of giving you this file is so that you have a reference when you are putting together your lab reports. Okay, this tells you what I'm looking for in terms of my expectations for how much you write and what you write in each section of the lab report. So we're going to go through that. We're going to go through how many marks each section of a lab report is typically worth. Okay, all of that kind of stuff to give you an idea of what you're going to be expected to do. Okay, that gives you the ability then to kind of target your work towards the mark that you want. Okay, if you're wanting 100%. Okay, then you know what you got a gun for. If you're happy with getting 75, then you know how much you got to put in. Okay, that's up to you. All right, but I'm going to show you kind of what my expectations are, okay, around how much I expect to see. Okay. So what we start out with, obviously, on any experiment is a problem. On any lab report template that you get, it'll say problem, and then it'll be blank. Okay? The expectation for the problem is that you write it out in the form of a question. So it's kind of like Jeopardy. Okay? You have to have what it is you're investigating in the form of a question. It's usually a very short sentence. Okay? The problem you're investigating is never something very long. Okay? The example that we're going to work with in our kind of run through today is very simple. What is the effect of light on plant growth? That would be a very simple problem to investigate, but that's what we're going to be running through as kind of our example here today. Okay, so the problem is stated as a question in your own words. Now, to give you an idea of what a lab report template looks like for you, okay, when you get, so this is your Properties of Compounds lab report. Okay, this is the file that you will work out of. Okay, you can see problem, and it just says restate the problem in your own words, obviously in the form of a question. So you just delete that and type in your problem. So all of your lab reports will look like this. I will have already built the template. Okay, so the bones are here. You have to put the flesh on the bones. Okay, essentially is what you're doing with these. So, okay, where it says manipulated, you would fill in what the manipulated variable is and why. For the responding, what is it and why. Controlled, what it is and why. Basically, you're just filling in everything, right? Anything that's already typed, like the materials, you don't need to mess with. It's just there for you. Okay, filling in your data. So I usually would have data charts already built for you to fill in. Okay, your analysis, conclusion, you fill it in. Evaluation, you'd fill it in. Okay, and then that's it. Okay, so that's what a typical lab report would look like before you do anything to it. Right? The template is built. All you have to do is fill it in. Okay, so we said here, the problem we're investigating, what is the effect of light on plant growth? Now, we kind of already know that, right? Do plants need light to grow? Absolutely. All right. It might be a little more complex than that, but we're going to kind of run with that idea. So the next step is the design. And in the design is where we identify the manipulated variable, the responding variable, and 3 controlled variables. Okay? So this part is out of 5 marks. Right? 1 mark for each variable. So the manipulated variable is the part of the experiment that is being altered on purpose by the experimenter. Okay? In a lab where I'm studying the effect of light on plant growth, what would my manipulated variable be?
The amount of light given.
The amount of light. Okay? I'm trying to study the effect of light on plant growth, therefore I, as the experimenter, need to manipulate the amount of light. All right, does that make sense to everybody? Okay, so what I would expect when you write out the variable is okay? Obviously, it's the part of the experiment changed on purpose, okay? And in this case, it would be the amount of light given to plants because all subjects will receive different amounts of light and light powers photosynthesis. So I've identified that the manipulated variable is light, and I've stated how and why it is being manipulated. That's what has to go in for you, right? So anytime you're listing out the variables, you'll say, this is the manipulated variable, and in your explanation, how it's being manipulated and why. So it says in here, different amounts of light will be given to plants, and light powers photosynthesis, that's the why. Making sense? What I don't want to see is the manipulated variable is light. End. Okay, that's not good enough. That's just identified, not explained. If you were to identify 5 variables correctly and not explain any of them, the best you can hope to get is 2 out of 5. Okay? The variables have to be explained as well. Questions so far? For our responding variable, some experiments will have only one responding variable. Some will have more than one. This one we're going to say just has one responding variable. And that responding variable will be the thing that we are actively observing, measuring, studying, okay, in the experiment. So in this experiment, the thing we're watching, measuring, studying is the growth of the plant. That is going to be our responding variable. Right? So the responding variable is whatever changes as a result of the manipulations made by the experimenter. So in this one, the responding variable is the growth of the plants, because plants getting more light have more energy for growth. Okay? The other thing that should probably go in there is, how are you basically measuring it? Okay? Growth of plants is pretty general. Right? What's a good measure of the growth of a plant? Could be height, could be counting the number of leaves, could just be taking pictures of it every day and doing kind of qualitative stuff. You could be putting it on a scale every day and taking its mass to see how much heavier it's getting, whatever, but you have to have some measure of your responding variable. Everybody all right with that? Okay, so identify and explain. How is it responding to the manipulated variable? Okay, and in all of ours, you need to identify 3 controlled variables. Now, controlled variables are variables that could also cause a change in the responding variable. We don't want that. We only want the thing we're changing on purpose to cause a change in the responding variable. So these other things have to be controlled, that is, kept the same throughout the experiment so that they don't have any effect, or their effect is equal for all of the subjects. So what are some other things that could affect the growth of a plant?
Amount of water.
Amount of water.
Like how nutrient soil is.
Yeah, the soil.
Its general environment and pest control.
Okay, yeah, and basically anything else in its environment. So, the temperature of its environment, if there's any bugs or anything like that. Okay, all of those things could affect the growth of a plant. So I want all of the plants in my study to have those things be given to them in equal measure, or for the condition to be the same. Okay, so controlled variables, other factors that could affect the outcome of the experiment if left unchecked. So what I'm looking for is identify, explain how and why it's being controlled. Okay, so how are you controlling it? That is, how are you keeping it the same? And why are you keeping it the same? In other words, what effect would it have on the experiment if you didn't? Okay, so one example here, same species of plant. It would hardly be a fair experiment if my subjects were a carrot, a bean plant, a rose, and a lodgepole pine. Okay? These plants are not at all the same. Okay? They all grow at different rates. They're all affected by things differently. I don't know how I get them all in the same size pot. Okay? Like it would be a terrible idea to not test exactly the same plant. Now if I planted like 5 carrots, okay, that would be a more controlled experiment. Okay? Or 5 bean plants or whatever. Okay? So, same species of plant because different plants grow at different rates. So, I'm saying I want to have the species the same. I'm making sure it is. And Okay, here's why. Different plants grow at different rates. Okay, the same amount of water. Okay, so another controlled variable is the amount of water, because water is an important factor in plant growth, so each plant needs the same amount, so it can't affect the growth rate of the plants. How and why? Okay, both explained in there. Okay, the same soil, because plants get nutrients from soil, and different soils hold water differently. Keeping them the same ensures it cannot affect the experiment. Okay, I actually have 4 in here. Same temperature, as plants typically slow their growth in extreme heat or cold. Choosing an effective temperature for all plants ensures it does not affect the experiment. Every one of those, how I'm controlling it, why I'm controlling it. Okay? Now, in the why, you do need to be specific. I get a lot of people who say, I'm keeping it the same because it'll affect the experiment. Yes, that's true, but it's not very specific. Okay? You need to be detailed when you're explaining that. Level of detail is important. All right. Any questions on the design part of the experiment? Okay. So every time we have an experiment, okay, and you open it up in Google Classroom, you'll see the scoring rubrics down below. Right? So these are the areas in which your lab is going to be marked. The design is out of 5. Your hypothesis, out of 5. Your procedure for this one, out of 5. There isn't normally a procedure, but because on this one you're actually writing the procedure, it's worth 5 marks. Your data and observations, 3 marks. Your analysis, 5 marks. Conclusion, 4 marks. And evaluation, 3 marks. So what I'm looking for here are the scoring rubric that I'm using for your variables. Okay? If you want to get 5 out of 5, all key variables are identified and explained. Problems clearly stated in the form of a question. Okay? 4 out of 5, most key variables are identified and somewhat explained. Okay, you see in the qualifiers that are different here. Okay, a partial problem is stated in the form of a question. Okay, 3 out of 5, some key variables are identified and poorly explained. The problem is vaguely stated in the form of a question. Okay, 2, few key variables are identified and not all are explained. The problem is incorrectly stated. And 1, little or no key variables are identified and are not explained, and the problem is not stated in the form of a question. And 0 means you just didn't do it. Okay. All right. Now, the next part of your experiment, your lab report, is your hypothesis. A hypothesis, you've probably been told up until now, is an educated guess. That's a terrible way to explain a hypothesis. In science, you never guess. Okay? Guessing is alchemy. In science, we want to have deductive thinking. We want to have a reason for doing something. We don't guess. So a hypothesis is always going to be an if and then statement. The if part is going to talk about the premise that we're working on. It's going to refer to the problem and how we think it would be solved. The and part briefly describes the experiment, very briefly, and the then part predicts our results, okay, or tells what we're going to be able to do with the data we acquire. Okay? So it works like this. If this is true, and an experiment like this is run, then this is what will be found. Okay? A hypothesis sets up so that it must be either right or wrong. Okay? And it is absolutely okay if after your experiment, your hypothesis is proven to be incorrect. Okay? You're not going to lose marks in your hypothesis if at the end of the experiment you have to say, I reject my hypothesis. That's okay. Most hypotheses in real science end up being rejected, and then a new experiment is performed that's slightly modified, and other things are discovered. It's totally okay. Okay? I always have people who they're going back and they're fixing their hypothesis because they didn't want to be wrong. It's okay to be wrong. Okay? I mean, most of the time for us, our experiments are pretty book, like, we're not gonna be wrong very often. But if you end up being wrong, that's okay. You just have to explain why in your conclusion. But it's perfectly fine. You're not gonna lose marks if your hypothesis ends up being proven unacceptable. Okay, so a hypothesis is an if, and then statement. So it has to contain those three words. Okay? I see this happen all the time in hypotheses, and it just annoys the heck out of me. Okay? Someone writes down for a hypothesis, I think this will happen. Well, goody for you. I don't care what you think, I care what you can prove. Okay? And a hypothesis is something you can prove. You can't prove what you think. Okay? You never say in a hypothesis, I think. Never. Okay? As soon as I see those words in a hypothesis, I'm like reaching for the 1 button, okay, out of 5. Because I don't want to know what you think. Okay? I want to know what you can prove. So it's an if, and then statement that communicates what the relationship between the manipulated and responding variable is thought to be. That's the if part. So for this experiment, it would probably need to say something about how light affects plant growth. Okay? Briefly describe the experiment, that's the and part, and outlines the expected results. That's the then part. Okay, so for this experiment that we're running, because it's very simple, our hypothesis is going to be pretty short. If plants require light for growth, all right, I just said, manipulated variable, responding variable. Plants need this stuff to grow, right? So I've covered that in the if part. That's a very simple if part, okay? And 5 identical plants are given different amounts of light. That's a brief description of my experiment, okay? Then the plants that receive the most light will show the most growth, okay? I've said what the relationship is. I've briefly described my experiment. And I've stated what I think my findings will be, okay? Or predicted what my findings will be. Is it okay if I'm wrong? Yeah. And I'm going to be, because that hypothesis is going to be proven wrong. All right, so the key things here, the if part, what's the cause of the problem, or the relationship being investigated. Okay, in this case, the relationship is light is what plants need to grow. Okay. The and part describes the experiment and then what to expect if you are correct. The other thing, we generally try to avoid the use of I, us, or we in a hypothesis. This is a scientific deductive statement. It should not include references to specific people, like I, me, or us. You'll notice that in my hypothesis here, I never refer to a person. I say, if plants require light for growth and 5 identical plants are given different amounts of light, then the plants that receive the most light will show the most growth. Never use I, us, we, ever. Okay? But it's very common for me to see that. Okay? Lots of times people will say, if plants require light for growth and we give 5 plants a bunch of different light, then we'll find that. No, you don't need to say the we. Okay? Just make the statement. All right. Everybody understand how that hypothesis is going to work? That's usually the part people struggle with at first, and that's okay. We'll get through it. All right, so the rubrics for the hypothesis, okay, look like this. So 5 out of 5 for a hypothesis is a clear, relevant, if and then answer to the problem with a relevant explanation or justification that uses concepts from class. Okay. For 4 out of 5, a relevant, if and then answer to the problem with a weak explanation or justification that uses concepts from class. Okay. If it's 3 out of 5, it's an unorganized but relevant answer to the problem with a weak explanation or justification that attempts to use concepts from class. Okay, and you'll notice that it doesn't say anything about it being if, and then. So if you're missing any of those, you're kind of starting to go into this territory. Okay. 2, a superficial answer to the problem with a weak explanation or justification that does not use concepts from class. Okay. A vague and or irrelevant answer to the problem with an irrelevant explanation or justification that does not use concepts from class. That would be like, the plants that get more light will grow better. Okay? If you wrote that as your hypothesis, that's a 1. Okay? And then obviously 0 would be not done. Okay? All right. Questions there? Okay. So your procedure, whoops. Okay, your procedure is going to be a detailed, step-by-step set of explicit instructions for preparing and performing the experiment. So for this first lab, you guys are actually going to be writing out the procedure. Okay, it's the only one where that will be the case. On the rest of them, the procedure will already be written for you by me, and you just have to follow it. Okay, so normally there aren't any marks for the procedure. Okay, just on this first lab. Now, when I say it needs to be step by step and explicit, I don't mean it contains a whole bunch of 4-letter words. Okay, that's not what I mean by explicit. I mean it is clear and leaves no doubt. That's what explicit means. Okay. So it also needs to be step by step. When you write a procedure, it needs to be a numbered list. Step 1, step 2, step 3. I see people write their procedures as a paragraph all the time. If I'm trying to follow instructions that are a paragraph, I end up having to read the whole paragraph every time I need to go to the next step because I can't find where I left off. If it's a numbered list, I know I just did step 3 and the next one is 4, right? It's easy for me to find where I left off and continue on from there, okay? We never want to have a procedure be a paragraph. Okay, observations. You do get 3 marks for your observations and data. So you want to make sure you do a good job of recording, okay, or measuring, or whatever is involved in a particular experiment. So for the experiment we're doing on Monday, it's just going to be making observations of color, solubility, odor, all that stuff we talked about yesterday. Okay, so you're going to have either a chart, or you're going to have graphs, or you're going to have pictures, okay, whatever it happens to be. When we get into biology, you're going to be taking pictures of stuff through the microscope, and then you're going to be labeling it. Okay, that would be your observations, right? We do an experiment where we're looking at water traveling through a plant. You're going to be taking pictures of the plant every day and kind of recording the difference. Observations come in lots of different forms. What I'm looking for is the quality of your observations. And if you've taken 2 pictures of a plant, day 1 and at the end, and they're so terrible and far away that I can't see any difference, that's not going to be a great set of observations. If you've taken pictures of the plant every day for 5 days from different angles, close-ups and stuff like that, that's different. That's a much more thorough set of observations. Okay? If you're recording lots of measurements, okay, in a physics lab, great. In the chemistry lab, if you just write white, okay, that's not great. White, crystalline, solid, strong odor, blah, 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 more observations, that's better. Okay, everyone following what I'm looking for there? Okay, so in the rubric for observations, okay, results are very carefully and very neatly recorded. Detailed charts/drawings are in proper format. Okay, results are well recorded. Detailed charts/drawings in partially proper format. Results are sloppily recorded. Detailed charts with drawings with improper format. Okay, that's what we're looking for there. Okay, your analysis. Your analysis is going to be out of 5 marks. And again, analysis varies from lab to lab. In this first lab, it's just a chart. And in that chart, you're telling me substance A is ionic, and it's this. Substance B is molecular, and it's this. That's all of the analysis in this first lab, is just finding the identities of the unknown materials. In some labs, it'll be 3 or 4 questions that you have to answer. Okay, so I'd be looking for complete sentences, thorough answers that incorporate what we went over in class, okay, that kind of thing. So they could be calculations. When we go into physics, it'll be, here's a graph that was built of the data, and here's the analysis I did of the graph. Okay, things like that. So answer questions, perform calculations, looking for detailed, explanatory answers. Okay, and then an answer to an analysis question that is yes, or no, or nothing happened. Okay, those are not good answers. Okay, those are not going to get you good marks on your analysis. Okay, so if we're looking at 5 out of 5, answers to questions show an in-depth understanding of the concepts of physical properties. That's for this lab in particular. Okay, answers draw insightful conclusions, and observations are referred back to when explaining answers. Also very important to refer back to your observations. Okay, all answers are very well explained in terms of concepts used in class. So if you were talking about, ionic compounds and some of their properties or something like that. Okay, extremely solid reasoning is used to defend answers. All compounds are correctly identified. Okay, 4 out of 5 answers to questions show an understanding rather than an in-depth understanding. Okay, and so you see just the qualifiers change as we go along. Okay, we're getting to the lower ones. They're showing partial understanding. Answers draw poor conclusions. Okay, few answers are well explained, weak reasoning, few compounds are correctly identified, sliding scale. Okay, that would do that. Okay, for your conclusion. Conclusion is out of 4 marks. What I'm looking for in a conclusion is, number 1, copy and paste your hypothesis. So just go right up to the beginning of your lab, highlight your hypothesis, copy, paste. Okay, that's the first thing. Then, based on your data, your analysis, explain to me whether or not your hypothesis is acceptable or not. Okay, is it acceptable or not? So for this lab that we did with the plant growth, let's say that my data showed this. I had my 5 plants. 1 plant was in the dark completely. Okay, never got any light. Second plant got 6 hours of light per day. Second plant got 12 hours of light. Third plant got 12 hours of light per day. Fourth plant got 18 hours of light per day. Fifth plant was 24 hours a day light. My data is that the first plant and the last plant both died. So the plant with 0 light and the plant with all light died. Okay? The plant that did the most growth was the 18-hour plant, and the second biggest was the 12-hour. Okay? Is my hypothesis supported by that data? It is not. My hypothesis said the plants that get the most light will show the most growth. That is unequivocally wrong. Okay? My 24-hour-a-day plant died. Okay? My hypothesis is not acceptable. So what I would have to say in my conclusion is something like this. As it was found that plants receiving small amounts of light and very large amounts of light showed little growth or died, okay, it can be concluded that plants require certain amounts of light and darkness to be healthy. Therefore, the hypothesis is rejected. So I talked about my data, I said what I thought it meant, and rejected my hypothesis. Okay? Everyone follow me there? The last thing I want to see is I was right. Goody for you. Okay? No, you weren't. You never say I was right in a conclusion. Okay? You can say the data seems to support the hypothesis. That's fine. Okay? But we never say I was right or I was wrong. Okay? We explain. Here's my data. Here's what the data says. Here's why I can accept or why I must reject the hypothesis. Okay, so if we're looking at the rubrics for the conclusion, okay, gives a valid conclusion that accepts or rejects the hypothesis based on correct interpretation of the results with a correct in-depth explanation, and where appropriate, compares results with literature values. Okay, 3 gives a reasonable conclusion that accepts or rejects the hypothesis with an explanation that has some validity. States a conclusion that has little validity or a weak explanation, draws a conclusion that misinterprets the results, an inaccurate explanation, and then not done. Okay. The last part of your lab report is your evaluation. This is your sources of error. So what I'm looking for here is for you to identify 2 things in the experiment that could have affected your results and how you would fix them. Okay. The way you fix them doesn't necessarily have to be something that we could actually do here. you can just say, this experiment would have worked way better if I had an electron microscope and millions of dollars. Okay, I mean, obviously I'm not looking for something like that ridiculous, but okay, like you can say, look, there was way too much friction in this lab. If I had, a air table and a bunch of fancy equipment that I could have eliminated friction, I would have got better results. Okay, for this particular experiment, okay, I would say that one source of error, okay, could be that the temperature was not continually monitored. Maybe we're in a room where the temperature goes down at night. Okay, now it would have been the same for all of the plants, but you said temperature was a controlled variable with it, which means that it should have stayed exactly the same throughout the entire length of the experiment. Right? So that could be a source of error. How do you fix that? You make sure that the plants are in a climate-controlled room where the temperature does not fluctuate at all, because that could affect plant growth. Another thing could be that the seed, the plants were seeded instead of grown from genetically identical cuttings. Okay, that means that you could have got one seed that was maybe genetically inferior to another and just didn't grow as well. That's a source of error. How do you fix that? The next time you run the experiment, you make cuttings from the same plant. Then you know all the plants are genetically identical, right? And that eliminates that as a source of error. So that's what I'm looking for. These things could have affected my experiment. Here's how they could have affected the experiment. Here's how they'd be fixed in the future. Okay. All right. Questions on the report, expectations, and format? Okay. So now you're probably looking at that and going, geez, Coderre's got really high expectations for his lab reports. I'm going to be spending like hours doing a lab report. No. Okay. I mean, I have high expectations. I won't argue with that. But a lab report should not be hours and hours of work. Okay? A good lab report shouldn't be more than, at the most, an hour's work. And that would include the time you spent in class. Okay? This first one's going to take you longer, but that's why we're going to have class time to work on it, because it's your first one. Okay? But once you get the hang of how to do them, you're going to get some class time to work on them. Okay? But you shouldn't be looking at, I've got to set aside 8 hours to finish this lab report for Mr. Coderre. If a lab report is taking you like an hour and a half to 2 hours, then you need to come and see me because you're doing something wrong. Okay? Or you're just putting in way too much work. Because I've had people, every time they hand them in, they're happy to put in 2.5 hours worth of work. And their lab reports look freaking awesome. Okay? But I don't want you to spend that much time. I want you to have a life. Right? You're a kid. You should be able to do other things than write lab reports. I still need lab reports from you, and I need them to be good, but I don't need you to spend your whole life working on them. Okay? So they shouldn't be hours and hours of homework for you outside of school. If that is not my intention, and if that is happening, then you need to come and see me so we can get it worked out. Okay? Because I have had that happen. I've had kids coming to me, and I've spent 8 hours on this. I'm like, what were you doing? Okay, like, did you fall asleep for 3 hours in the middle? Like that, like, I don't know, like, what are you counting as the time you actually spent on this thing? It shouldn't take that long. All right, questions there? All right, I'm gonna give you guys, let's say 9:25, we're gonna come back and we're gonna start looking at the properties of compounds pre-lab stuff. Okay, so I'll give you a 5-minute break, and then we're gonna do that.